Hi everyone, my name is Magda Bienko and I'm an assistant professor at Karolinska Institute in Stockholm, where I lead a lab for quantitative biology of the nucleus. And today it is my great pleasure to introduce to you our most recent technological developments with which we are trying to advance knowledge on how our genome is organized in the 3D space of the nucleus. In my lab, we are after this seemingly simple, in reality very complex biological problem, and that is how the 2-meter long molecule of DNA can fit in the tiny volume of a mammalian nucleus compatibly with all its functions. By now we know that our genome is not entirely randomly organized. There is some structure to it for sure. We know that, for instance, individual chromosomes need to come together for nucleoli to form. We know that the centromeres tend to cluster, telomeres cluster as well, and that certain genomic regions have this strong tendency to be associated with the nuclear lamina, to form lamina associated domains. Individual chromosomes occupy discrete volumes in the nuclear space called chromosomal territories, and at least in some organisms, chromosomal homologs tend to pair or at least be located at certain reciprocal positions. And as I mentioned before, there's a big chunk of DNA that needs to fit in this small volume, so there's the problem of molecular crowding. And so all in all these constraints lead to a certain arrangement of chromatin in 3D, but we are definitely far away from understanding it all. We don't know still, for instance, how individual arrangement of chromosomes, subchromosomal regions, how they differ from one cell to another, from in between different tissue types, let alone what are the driving forces that lead to a certain arrangement of chromatin in 3D. And so in recent years, my lab has been developing various methods, various tools and technologies to study chromatin architecture. We have been developing various microscopy approaches, mainly DNA and RNA FISH, to look at individual topologies of DNA and how they relate to gene expression. We have been studying, using DNA FISH, details of chromosome structures, their arrangement in 3D, and basically looking for various spatial patterns of their localization. And more recently, we have been developing various sequencing-based approaches, and today I'm going to present to you one of such approach, which we called GPSeq. So GPSeq stands for Genomic Loci Positioning by Sequencing. It was developed in my lab by Gabriele Girelli, Tomasz Callas, and Joachim Custodio. And I always use this analogy to an onion for this method, because here what we are doing is sort of peeling the nucleus like an onion, trying to understand what DNA sequences tend to sit in which of its concentric layers, basically. So in GPSeq, we have developed a protocol for gradual digestion of fixed chromatin by a restriction enzyme. We rely on gradual diffusion of a restriction enzyme through a meshwork of fixed chromatin in cells, whereby if we treat our material for a very short duration of digestion, the enzyme should digest only the very outer layer of chromatin. If we let the cells sit for a longer period of time with such an enzyme, the enzyme will go a little bit deeper and digest a thicker layer of chromatin, always starting from the periphery, and eventually it will go all the way to the center of the nucleus, and the whole volume of the nucleus will become digested. We obviously wanted to make sure that we can visualize, we can monitor the enzyme digestion to make sure that the enzyme is actually doing what we wanted it to do. So we developed this new FISH approach where we ligate this Y-shaped linker adapter to the digested sites, and then we are hybridizing short fluorescently labeled oligos to this linker, to this adapter, in order to visualize which of the cut sites were digested by the enzyme. And so what you can see here, this is what we see under a microscope. In green is our whitefish signal. That's where the digestion happened. So after 10 minutes of digestion, the enzyme will only cut the very outer layer, 15 minutes a little bit deeper, 30 minutes even deeper, eventually really filling in the entire nucleus with the cuts. Now, if instead of ligating Y-shaped linker, we ligate a linker that supports sequencing, we can generate sequencing libraries from these individual time points. And when we combine the sequencing data all together, we generate for each locus so-called GPSeq score that tells us about its radial location, how distant it is from the nuclear periphery or the nuclear center. And so here I'm showing you a distribution of such a GPSeq score from human HAP1 cells. You can see that the profile differs quite a bit all along individual chromosomes, as much as in between individual chromosomes. The higher the value, the more central location is, the lower the value, the more peripheral. This map is in 1 megabase resolution, and we get very high reproducibility at 1 mega, which stays still very high even at 100 kb resolution, and now we can even go down to 25 kb resolution. We obviously wanted to validate this approach using orthogonal methods, and our gold standard is the 3D DNA FISH that allows us to measure distances in single cells, directly in cells under a microscope. We used 68 randomly selected DNA loci, 8 kb loci, scattered all throughout the human genome, and we measured their distances, the typical distances from the periphery, and we were very pleased to see that these distances correlate very highly with what we get from GPSeq at 1 megabase resolution, as well as at 100 kb resolution. And so basically at this point, this gave us a green light to proceed to further analysis, being confident that what GPSeq reveals to us are true radial distances. And so what can we use GPSeq score for, or maps of radiality? We were very keen on adding this radial dimension to the study of various genetic and epigenetic features of chromatin. So we combined GPSeq score with various omics type of data. For instance, we used ATAC-seq data to study radial distribution of DNA accessibility in the nucleus, in the human nucleus. We divided the nucleus into 10 concentric layers, with 1 being the very periphery and 10 being the very center. You can see with this dashed line that DNA accessibility, as expected, goes up towards the nuclear center. This is where most of the transcription is taking place, so this comes as no surprise. What we did on top of that was to stratify this analysis by high C subcompartments, so different chromatin types, A being the most active, so U chromatin, B being heterochromatin or the B compartment, with B1 being the polycomb repressed chromatin, B2 pericentromeric, mainly with some regions belonging to the lamina-associated domains, the nucleoli-associated domains, and B3 being enriched with LADs, lamina-associated domains. And so what you can see here from such analysis is that, for instance, when you look at regions belonging to the B1 subcompartment, so polycomb repressed, they actually show higher DNA accessibility if they are placed in the very center than some of the A1s, so the most active chromatin, if that is placed more peripherally. So it really shows you that this radial dimension adds a lot to the picture. Looking at various epigenetic features along the radius in different subcompartments, we really got this impression that these radial profiles change a lot from one subcompartment to another. So, for instance, looking at H3K27 acetylation, again, as expected, genome-wide with this dashed line, you can see it increases towards the center. But when you look, for instance, at the B2 subcompartment, so pericentromeric, for instance, it actually goes down towards the center, this mark. when we looked at the inactivating mark, H3K9 trimethylation, we could see opposite trends. So here, genome-wide, it seems to be going at least slightly down towards the center, but again for the B2 subcompartment it looks very different. There is a sharp increase of this mark towards the center, and that basically made us think of heterochromatin that is present on the small chromosomes like chromosome 17 or 19 that are known to be present typically in the very center of the nucleus. They do carry heterochromatin, they have pericentromeric regions, but these regions are exposed to very highly active sort of transcriptionally active environment, and so perhaps they really need this boost of inactivating mark and a depletion of activating mark. What we also looked at was whether genes that belong to a common pathway show any peculiar radial behavior of their distribution. And so what we saw, this was particularly interesting for us, was we saw that basically genes that are responding to UV irradiation, if they get upregulated upon UV, they tend to cluster in the center, while if they are downregulated upon UV, they show the opposite trend, accumulating more towards the periphery of the nucleus, which we find really interesting. We also looked at distribution of transcription factor binding sites, and we could see that for the vast majority of transcription factors, their binding sites show actually strong radial gradients. So either anti-correlating or correlating highly with GPSeq score, indicating again some functionality to it. And that is something that we are now trying to explore in greater detail. Now, when we developed or already when we conceptualized GPSeq, it occurred to us that having these highly accurate radiality maps could be very useful in genome reconstructions, or basically in using data from Hi-C method that reveals intra-chromosomal contacts and basically placing these contacts properly in the space of the nucleus. And so Erik Wernersson, a senior scientist in my lab, developed an algorithm which he called ChromFlock that combines GPSeq data with Hi-C data. It's based on the PGS software from Frank Albert's lab that uses Hi-C only. And basically, the algorithm generates thousands of single instantiations of genome structure, either at 1 megabase resolution or at 100 kb resolution. Still, even at 100 kb, recapitulating basically the expected pattern of arrangement of various subcompartments. When we looked at our structures from ChromFlock, we could see that they correlate very highly with our GPSeq maps, as expected, since we are using them to generate the structures, and they correlate very highly with the original Hi-C maps, again as expected, sort of a proof that we are doing something right here. But when we generate such structures using Hi-C only, they actually do not correlate very well with our radiality method. So we believe that they do not preserve, or Hi-C might not be enough to position properly contacts in the space of the nucleus. And even when we look at correlation with the Hi-C data, the original input data, the correlation looks worse than when we use also GPSeq to generate these structures. Most importantly though, when we look at correlation of these structures with the 3D DNA FISH, we see also a very high correlation for our structures when we use Hi-C and GPSeq, and no correlation really when we use Hi-C only. We went a step ahead further and basically wondered what would happen if we entirely omitted trans contacts from Hi-C. So really focusing only on intrachromosomal contacts, taking only those with GPSeq. What would happen if we generated 10,000 structures there? And so, as expected, again, they correlate very well with the GPSeq. They actually correlate equally well with the original Hi-C data that has both trans and intra contacts. And to our big surprise, but also big satisfaction, of course, they correlated very highly with our FISH data, again suggesting that that GPSeq does a really good job in positioning contacts properly in the space of the nucleus and doing a good job in aiding in the reconstructions of the genome. When we obtained our radiality maps, given the fact that here we have sort of data all along the radius, GPSeq allows us to study radiality all across the radius, not only looking at what is at the very periphery versus everything else, or what is in the very center versus everything else. We really profile everything all throughout the radius. And so we were very keen on using our data to test the long-standing so-called bodyguard hypothesis proposed back in 1975 that says that the very outer layer of chromatin, heterochromatin sitting at the very periphery, acts as a shield protecting euchromatin from insults by mutagens that would enter the nucleus from the outside. And so to address this, we looked at the distribution, radial distribution of of SNVs, single nucleotide variants found in different cancer types, and basically looked for their density in different radial layers, and we could indeed see that they do accumulate, they are enriched at these peripheral layers in comparison to the more central ones, especially pronounced for cancers such as melanoma or lung cancer. On the other hand, when we looked at radial distribution of endogenous DNA double-stranded breaks, we could see that those tend to accumulate more in the very center of the nucleus. And this made us wonder, or made us think about a related phenomenon, basically gene fusion found in cancer. And because we know that double-stranded breaks are required for gene fusions to occur. And so we wondered about where genes, which are found frequently being engaged in gene fusions, in chromosomal translocations, where would those be located, radially speaking? And indeed, we could see that also in this case, they do accumulate, or they are enriched in the very center of the nucleus as compared to regions that do not carry such genes. And we moved again step further, digged a little bit deeper. Thanks to the fact that we have chromflux structures, we could actually ask more questions, sort of going into further mechanistic details of gene fusions, and asked a question of whether beads in our structures that carry genes engaged in gene fusions would mingle more. We hypothesized that such regions would be engaged much more in interchromosomal mingling, and so we asked exactly that, and we could indeed see from our chromflux structures that beads which carry genes engaged in gene fusions, they are much more frequently engaged in interchromosomal mingling events. All of this basically pointing to certain mechanistic details behind chromosomal translocations formation, for instance. So sort of we like to think that this approach is really good for studying the interplay between genome structure and genome fragility. And with this, I basically summarize my talk, giving you some take-home messages. We have established a protocol for gradual penetration of proteins through chromatin in morphologically preserved cells. I showed you this for restriction enzymes, but we tested also for antibodies, for instance, and that works really well. GPSeq allows us to generate highly resolved radiality maps. Chromflux generates state-of-the-art genome reconstructions. Our data is in line with the bodyguard hypothesis, and the GPSeq serves the basis for new methods developments, and that is sort of ongoing right now in the lab, so please stay tuned. And with that, I would like to thank all the members of my lab, as well as all our collaborators, as well as all our funding bodies, and I thank you for your attention, and I'm happy to take questions. Thank you.